Good morning, everyone. So first, I would like to thank the organizers for giving me this opportunity to present my work. I work at the Charles Zuckerberg Biohub, where we are trying to understand zebrafish development. And for that, I developed this tracking software called ultrack, which I think it could be applied to several problems in tracking. So today, I'm not going to dig too much into the biology, but what we can do with this software. So when we started working with tracking, realized that what make tracking hard was segmentation, because when segmentation fails, tracking fails, or it's very hard to recover from it. So given that, we developed this software with a few features in mind. We wanted to be able to leverage existing models that people spend a lot of time annotating data, or you could use your own algorithm where you have more control of what it's doing. So we can use different sets of segmentation as input, build a set of segmentation hypotheses, and from that, we use temporal information to find the segments that are most coherent over time, capturing the final segmentation and the tracks. Our software, it also supports 2D, 3D, multicolor, or data sets that are larger than memory. And what I mean by supporting is going beyond just being able to use it, but we also provide some functionality that you're able to extend the existing algorithms you have to these different modalities. And also we have a simple Python interface. So if you have any format that is compatible with NumPy, Dask, or Zar, you should be able to use it to track your data without having to duplicate your data set. And to do that, we explore the relationship between segmentation and contours. So if you have a binary contour, you could fill each closed contour with a unique label and you'll be able to get a segmentation. If you have a segmentation, you mark the pixels that have neighbors with different values and you get a binary contour. And this relationship extends to multi-level contours. And a multi-level contour is equivalent to a hierarchical segmentation, represented here as a dendrogram. So what we do to get a segmentation from a multi-level contour, you can just think about binarizing it and applying the same scheme that I showed before. And it's equivalent to picking a threshold on the dendrogram of the hierarchy. And as you increase the threshold, you're merging segments into merged, and you have a single segmentation. So you form this tree structure. But what happens in practice is that a lot of times you need more than one threshold to get the accurate segmentation that you hope for. So it's not a horizontal cut in the hierarchy, and that's a very complicated problem. And what we do here is that with the temporal information, we can detangle and get the right segmentation, finding the tracks at the same time. So here I have an example where you have a 3D segmentation. Sometimes it's even by eye, it's hard to know if it's one or two cells when you are looking at a single time point. We have this intermediate hierarchical representation as contours of the data, and then we can do the tracking, assigning the elements through time. So how can we create this hierarchical segmentation? There are several ways. I'm going to show you one in this data set where you have some cells with varying brightness. And I think the first thing I tried was to correct the brightness by adjusting the gamma and to have it to be more uniform. And then I applied a off-the-shelf segmentation model, in this case, Alpose. But still, we had some mistakes on the segmentation results. I tried different gamma, and I was trying to tune to find the best parameter. And you can see the segmentation errors highlighted by red. But the thing is, there was not a unique parameter that achieved the results that we wanted. So what we can do with this framework is that we get the boundaries of all these segments. We get the average of them, creating this multi-level contour, and then we can track on this space, achieving a more accurate result. And even when, so this data set, we have the ground truth, and we were looking at the quantitative results, and we can see that this approach was the most accurate. And in practice, a lot of times you won't have ground truth data. So tuning this parameter to get the best segmentation is really challenging. You might not be able to do that. So using temporal information to find your segments can be really powerful. So to get more into the details, how we pick the segments and find the tracks. So we estimate the hierarchy for each time point in your data set. And then we compute an association score between the time points, between the segments in the hierarchy. And then we formulate an optimization problem where we are trying to maximize the association between the two segments while having some constraints so that a pixel belongs only to a single segment. So in this example, we picked segment A, B, and C. So once C is selected, CD is blocked, and the same thing is for AB. So AB is selected, segment A and B is blocked and cannot be part of the solution. And this is optimized globally, not from pair to pair. So we apply this methodology tracking the whole zebrafish embryo, where we have this recording of over 8 hours, generating about 3.5 terabytes of uncompressed data. And with this methodology, we can track the bulk of cells and we can conduct our further analysis, but also having accurate single cell tracking accuracy. And despite having the zebrafish embryo growing and the cells going deeper and deeper inside the tissue and this the cells getting more scattered, as you can see in the ZX slice, we are still able to track the cells. And this was computed with the command line interface where it's compatible with Slurm or Nextflow and other orchestrating interfaces. We also tracked a multicolor data set. And in this case, we got the data from a collaborator. We didn't have the multicolor segmentation model, and we didn't want to spend time annotating it. So what we did is that we split the multiple channels into their own videos, and we apply a off-the-shelf segmentation model to segment them as grayscale, obtaining separate segmentations. We noticed that the deep learning model was also missing some group of cells because of different intensities. So we just applied our own algorithm using threshold and watershed because we had much more control. And with these 6 segmentation labels, we can combine them in a multicolor contour map, as I showed before, and then we can track the cells. And if, despite having a very crowded environment, we can still have extremely accurate segmentation by using the color information. And as we dive deeper into a single cell, we can also see we are capturing the cells in their divisions. So how accurate is it, right? I think most of the examples that I show here were qualitatively. So we submitted our results to the cell tracking challenge, where we have some training data, but the test data is hidden by the organizers, so they're the ones computing the tracking results. And the score they report here is the score of the combined segmentation and tracking score. So for the C. elegans, there is some training data for dense annotation. So we have almost the annotation of all cells. So we train a deep learning model to predict the cell boundaries and ran our algorithm. But on the other data sets, we compared, we don't have the dense labels, just the sparse annotations. And we compared our deep learning model with a more traditional approach using filtering and threshold. And that approach was better. So that's what we submitted. And still with that, we were able to get first place in 2 out of the 3 data sets of large 3D data. And I would like to highlight especially the Drosophila one, where we have a very large gap with other approaches. And that's just using filtering and thresholding plus our tracking algorithm. And all of that can be done with very few lines of code. So here is an example how you could run that in Apari, in Python, and visualize the data in Apari. But also we have several Jupyter notebooks as examples. So you can reproduce what we did. And I would like to thank my teammates. All of this could not be done without them, as well the BioHub staff, including IT and computational microscopy, and our collaborators as well. Thank you for your attention.
Great, so we're doing quite well with time. So please, if you have any questions for Jordao, head to one of the mics, or I could attempt at throwing the throwable microphone, but I'd rather not. And if there's any questions online, then also. Yeah, go ahead.
Nice talk, thank you very much. I'm wondering, so basically tracking is really depends on the time steps that you have between different slices. It seems like your images, the time step was relatively small. What will happen if the time step is large? So basically cells are so skipping from here and there. Will that still be robust?
So yeah, that's one downside of the approach, but I think that can be fixed, and we do fix in some case with registration. And our package allows to apply diffeomorphic registration without changing the data. So if you supply a vector field with the movement of your cells, that is applied at the segment level, so it's very easy to use.
That was really impressive. So when we track cells, we often have like one label that is constant over the track and then other labels that change in color, like Fuji probes, for example. Is there still an advantage to have all color labels segmented?
Yes, so we as the multicolor example, we can use the color information to improve tracking. And I think in that case, you could also, as long as is a smooth transition over time, that should help segmentation, the tracking, because if you have cells that are right next to each other but in different states, they will not get mixed up.
Maybe I'll follow up on that. So again, if in the case of multi-label, if we're looking at 3D tracking, would it cope well if, let's say, our markers are on very different planes? So you would have a marker which is, for example, very apical, another which is very basal.
Yes, that should work because we average per segment for the hierarchy.
Great. So if there's no further questions, let's thank Jordao again.